Hello, everyone, and welcome to today's keynote webinar titled Closing the Gap, Solving Complex Medically Relevant Genes at Scale. This webinar is part of the 12th Annual Precision Medicine, Genomics, Genetics, and Molecular Diagnostics virtual event series. I am Kaylee Bach of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots. Before we begin, I would like to remind everyone that this event is interactive. We encourage you first to participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the bottom left of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Help Desk button from the lobby within the event. Finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits image located on the right of your screen and follow the process to obtain your credits. All right, now let's get started. I'd like to now present today's speaker, Medhat Mahmoud, postdoctoral associate. For a complete biography on our speaker, please find it on the left of your screen. Medhat, thank you for joining us. You may now begin your presentation.
Thank you, Kelly. Highly appreciate it. And thank you for inviting me today. Highly appreciate it. Thank you for the audience for attending. Today, I will be speaking about closing the gap, specifically solving complex medically relevant gene at scale. And before I go forward in my presentation, I would like to give you, in a glance, why I'm here and what is my focus. My overarching research focus is leveraging healthcare by empowering individuals with personalized medicine tailored for their unique genetic blueprint. And to be able to achieve such a thing, I classified into 3 points that we need to fulfill. The first one is develop algorithm and method for comprehensive and accurate analysis for individual genomes, followed by be able to detangle complex gene at scale with lower cost, of course, and doing both of those things we could lead to unraveling the interplay between genetic and lifestyle to help us predict and treat diseases more effectively. Before going further in my presentation, I would like to give you a brief introduction about sequencing in case if you are not familiar with the term or the technology that's applied in sequencing. We could classify sequencing into 3 eras: first generation, second, and third generation. The first generation, like Sanger, for example, which was used for completing the human genome sequencing, it characterized by highly accurate read with length of around 1,000 base pair. However, there is an issue here that the yield is low, which leads us to the second generation, which fulfill and solves this issue. For example, we are using here Illumina, which managed to parallel sequence the genome with a high throughput and high accuracy. And the read length was between 50 to 500 base pair. That's why we are calling it short read. However, this was also the downside of short reads. Specifically, if we have a stretches of the genome with repeats over 500 or 50, or we want to identify structural variation, which genomic alteration that are 50 base pair or more, which could be a duplication, deletion, insertion, or translocation. If the size of the structural variant is larger than the read length, as in short read, for example, it would be problematic to call it accurately and effectively. There are several papers that saying that around 50% or more of the structural variant we are missing if we are using short read actually. To overcome that, we had the third generation, like BacBio and Oxford Nanopore. The main idea of third generation is to produce a higher or a larger read compared to short read and still save the accuracy. There are 2 technologies that are dominating the market, BacBio and Oxford Nanopore. The first thing, they are actually sequencing DNA in real time, which give us the opportunity to actually pinpoint the methylation signal, which should play a crucial role in different disease. Additionally, the read length on average is 20 kilobase, surpassing what we have previously shown short read. Even for Oxford Nanopore, the read length could reach to 4 megabases, which help us dramatically to solve large stretches of repeat or GC content or repetitive sequence in the genome. Also, the error rate is not as equal as Illumina. It is higher, but still low at the same time. Meaning for BacBio, we have less than 1% error rate, and for Oxford Nanopore, we have less than 3% error rate. With that said, now you have an understanding of what is sequencing is, what is the generation that we are talking about. In my research, I will be focusing on long read or third generation, and I will explain why in a second. Also in this presentation, I will be highlighting two main points: rare diseases and complex medically relevant genes. So let's start with the first part, which is the rare diseases. Here I will be showing the GREGoR project, which stands for Genomic Research to Elucidate the Genetic of Rare Diseases. But what is a rare disease? It is any disease that are affecting less than 200,000 in the States. So far, we have around 7,000 rare diseases that we are aware. Additionally, there are around 3 to 5% of the population that are suffering from rare diseases. Overall, we could say that 72% of the rare diseases actually is caused by Mendelian inheritance issue. With that said, what is our goal in GREGoR? The first thing is to link rare diseases to their genetic causes. By doing so, we will allow an improvement in genetic testing. So if we know the phenotype and what is causing the phenotype, we could easily pinpoint now the cause of it or develop test kit for doing so. Additionally, we will shorten the diagnostic odyssey for families that are affected by rare diseases. Currently, there are 5 research centers and data coordinates that are working on such a project. Before going further, and specifically for a project like GREGoR, actually what we are missing to have such an initiative. First of all, most of the clinical sample have been sequencing using exome, or focus was using exome. But an exome did a great job in pinpointing a number of diseases and actually identifying what variant is associated or linked to a disease, which is great. However, in exome, we are focusing only to protein coding region, which represent 1 to 2% of the entire genome. Thus, we are lacking the knowledge in 98% of the genome. Besides that, exome could have poor coverage in some region we cannot capture. We are not aware of what is happening in non-coding region, which play a crucial role as a regulatory element. Add to that, there are some cases where we have exome is ineffective because of complex phylogenetic conditions, where different genes actually orchestrate the phenotype. Finally, as I mentioned in the first slide, structural variant, which is larger than the reads that are used for exome, single nucleotide variant in complex genomic region, and phasing and methylation are absent when we are using the exome sequencing. Putting all of this together show that while exome sequencing is important, it is not answering all the questions that we need to answer to be able to solve rare diseases. We put all of that together in a paper, we called it Beyond the Exome, where we discuss exactly the steps that we are taking in the clinical analysis, starting from exome sequencing and when we can use different sequencing methods like whole genome sequencing or long read whole genome sequencing. I encourage you to go through this paper. I believe now, by this introduction, you know my role in this project, in GREGoR specifically. My main purpose here is to solve the unsolved. And now I will take you in a short journey, explaining to you what kind of samples that we have, the number of samples, what we do to analyze them, and how we interpret the result. In GREGoR, we have more than 1,600 enrolled individuals. More than 400 of those are families, representing around 284 diseases or disorder. 76 or more of the data that we have in Baylor are in trios or more than trios. As we can show here in figure A, this showing the number of the trios that we have, which is more than 300, adding to that the samples that we have more than 3 members of the family, which is great because it gives us an opportunity to dive deeper in the sequence for all the family and be able to solve them in a coherent way. Additionally, as we can see here in figure B, we are actually covering span of disorders, including neurological disorder, cancer, skin disease, cardiovascular disease. And what is more interesting, that we are spanning a different background individual, ranging from Native American to white and other individual. So putting all of this together, it's important to see that we have a really high number of samples that representing more than 284 rare condition, 400 or more, or 76% or more of these cases are trios, which give us the opportunity to be able to solve them. And for such a project, we used long read. Specifically, we used Oxford nanopore to be able to solve these cases. Starting with the QC, or for the pilot studies that we have, we could see that we have a good coverage around 25x or more, as you can see here on the graph on the left-hand side. I would like to stress that this DNA is really old, so it's really hard to sequence and have a good coverage of that. Nevertheless, we managed to achieve the coverage and high read lengths, which help us, as I mentioned, in solve complex or repetitive region and identify structural variant correctly. Besides that, we have a max length more than 1,500 kb with a high N50, which also support our aim to solve the complexity of the genome. Besides that, we need a really good, robust mechanism to analyze our data. For that purpose, we have, as I mentioned, 40 samples. 12 of those are families and 3 are singleton. And I want to answer today how we analyze them. I started by developing PRINCESS, which is a framework for comprehensive detection and phasing of single nucleotide variant, structural variant, and detecting and phasing methylation. It works in workstation and high-performance computing platform, so it could work in parallel. On average, we could analyze the sample fully in less than 15 hours. PRINCESS started by the read, would give you a QC about the read, meaning number of read, quality, average read length, the coverage. So you have an understanding of how your sample looks like. Then it will begin by aligning data using Minimap. In developing PRINCESS, we focused on simplicity. So all it needs is just the read and what kind of data that we are analyzing. Either it's from Oxford nanopore or from BacBio. Then it could itself to select the parameters that's needed for each scenario, filtering data, so it will give a coherent SNVs and SVs and phased data. After mapping, we begin in parallel to call single nucleotide variant using CLAR, which is a deep neural network that was tailored specifically to cope with the errors that we have from long read. Besides that, we are calling structural variant using sniffles, and we are genotyping our data using WhatsApp, and also there are additional steps to use the parental data and call methylation. All of this is happening while we're giving QC for each step. So at the end of the run, you will be able to get a QC for all the raw read, aligned, and SNV and SCV calling, phasing the data, and all of this in less than 15 hours or so. And it's publicly available, so feel free to use it or contact me directly if you need any help. We apply PRINCESS on the samples that we have, and we begin to analyze them. Here I am showing 2 examples of one of the samples that we have in hand, where we compare head-to-head the structural variant and SNVs. So for SNVs, we are showing here in graph A, we are comparing the paternal data to maternal and proband. The aim here to identify what is shared and what is unique. As you know, we have around 5 million SNVs and indel per genome. So we want to actually reduce this number to be able to focus on what is unique to the proband, which could be the causative of the condition, which was around 4% here, but this number is high because it needs to go on further filtering process. For structural variant, which is figure B and C, again, we are calling the variant for the paternal, maternal, and proband, comparing them together, and try to pinpoint what is unique to proband to analyze it even further. Moving from this step, you have a really large number of SNVs, indels, and the structural variant. So it is important actually to annotate them and know exactly what they are causing or what they are responsible for, or if you need further analysis in this direction or not. To do that, we developed a workflow to annotate the data that we have. For a single nucleotide variant and indel, we begin by annotating them with the population allele frequency. So we can filter later by variants that have low allele frequency in population, which could be actually linked to some diseases. Besides that, we are annotating by coordinate and functional impact using SNP effect and other deep neural network tools like CAD and DAN, which give us a score about the deleteriousness of a variant. So if we have a higher score, meaning that this variant actually could be the cause of the case that we are investigating. For structural variant, it was a little bit challenging. First, structural variant is hard to identify in the first place. Second, the functional impact is complex to interpret. As you know, the structural variant is at least 50 base pairs, so it could be covering more than one gene or one regulatory element. So we did different things. First, we used SNV annotate to overlap with the allele frequency from population as we did with SNVs. But the issue is that most of the structural variants that we have in the population actually was based on short reads, meaning that we are missing a large number of structural variants. So actually, it will not help us that much. Besides that, we are annotating them to identify the function, which is functional annotation, and rank them. This is so far what we found. Specifically for SNVs, we faced an issue that I mentioned, around 93% of the uniquely identified structural variant lacking annotation, which actually directing us to the need of calling a structural variant in population using long read. And there are current projects that are working on this direction, like 1000 genome, but with long read, not with short read, and all of us. Additionally, we need a better annotation database that's specifically for structural variant. And actually, we managed to develop, we're currently developing one of those. Here in our lab, we are developing sticks. This work is done by Xinshang to annotate the structural variant by building a database of structural variant from long read from population, where we take a BAM file or CRAM file and create a k-mere of the data that we have from the read, and then use it as a database. As you can see here on the right-hand side, we are comparing the false positive rate to the true positive rate using different buffer or slope, and we find that 100 is better. Regardless, I would like to show you what we managed to achieve with this text. Here we are looking to two graphs, A and B, or left-hand side read, which we are comparing short read to long read. As you can see here, we have a high consistency between both, meaning that we still achieve a high precision. And looking to the right-hand side, where we are looking to the challenging medically relevant gene or genes that cannot be identified by short read correctly, we could see that there are lines on the y-axis of blue hexagonal dot, meaning that here using sticks, we managed to annotate the structural variant that we were not able to annotate previously using available database, which was crucial and helped us to reduce the number of SNVs that we needed to look at. Moving from this, now I would like to show you some of the cases that we are focusing on now. So far, you knew the workflows that we are using, how we are annotating them, what we were missing from annotation part, and now what we could achieve. We start with this case, which is CHORS syndrome, where we have 10 weeks old male with developmental disorder and several birth defects. We started by exome sequencing, but as I explained earlier, it was inconclusive in such situation. Overall, we have around 536 candidate genes, which makes our job is really hard to analyze all of these genes and pinpoint exactly what is the cause. Regardless, we managed here to identify a structural variant. If you are not familiar, this is an IGV screenshot. We have the first band where there is a blue large bar. This is explaining the deletion mark, and it's around more than 2,000 base pair deletion. The second banner, which titled by sun, this is the sun reads, and here we can see this dark blue bar, which is representing a deletion in the sun. We look to the mother, you have this gray bar with no black solid line, which is meaning that absence of this deletion and the father, which have a heterozygous deletion that may be inherited here in the son, or also the son have different conditions. So beside identifying this deletion, it is still unconclusive, meaning we need to do further research on this specific case to show if this is the cause of the disease or not. And we are still working on that. There are several samples. As I mentioned, we have more than 40 samples. But for the sake of time, I will just show you this case, and I will conclude this part at the moment. So what we learned so far. In this part of my presentation, I showed you the analysis workflows that we managed to develop and prioritization of variant. But what we are missing is integrating short tandem repeats, copy number variant, which we are working on currently to involve them in PRINCESS. We managed to build a robust strategy for sample analysis, which could work as a backbone for further projects, like I mentioned, 1000 Genome or All of Us, which will help us in identifying variant in a more efficient way and pinpoint what is the relation or the link between the variant and the phenotype. We show you the long-read application beyond single family to a cohort-wide, and I prove that we need to improve our annotation to be able to solve our cases. These all together will help us to achieve my ultimate goal, which is integrative long-read into clinical setting to pave the way for personalized medicine. Which will take me to the second part of my presentation, in which we will be talking about the complex medically relevant gene. So in the first part, I highlighted rare diseases. Now I will be focusing about complex medically relevant gene. But what are they, where they came from? What do we even mean by complex medically relevant gene? The term itself appeared in 2016 when a paper by Mendelker et al. Published in Genetics in Medicine, in which they are highlighting 193 medically relevant genes that are not fully accessible by short read for the reason that I mentioned earlier, but I will mention it again. But where the gene came from, why we cannot solve them, or what makes them complex. The genes have been aggregated from different databases like OMIM database or Human Genome Mutation Disease Database or ClinVar. The characteristics of this gene makes them complex to resolve because of low mapability arising from high or low GC content or repetitiveness. It did not stop here, actually. In 2022, actually, we expanded this number of genes to be now 386. I would like you to remember this number because I will use it in a different part of my presentation. So now we could say that we have 386 genes that are linked to several diseases, but we cannot solve them. Technically, we are blind there. This cartoon shows you how this genes was aggregated. As you can see, we use different databases I mentioned in the previous slide, OMIM, HGMD, and ClinVar, plus other data. We count and take the unique coordinate of the gene that are medically related. We have around 5,000. And we look to this gene in Genome Reporter, which is a benchmark data set of how many genes we could solve. We found that there are 395 genes that are not solved in both GHR37 and 38. So if we look only to this 38, which I'm using in this analysis, is 386, which I'll be using further. We took the genes that we identified as challenging and applied in a different project called All of Us. In a brief, All of Us is an NIH initiative that aim to tailor medical treatment for individual, accelerate health and medical breakthrough, and enable individualized prevention, treatment, and care. They are doing that through identifying risk factors for certain diseases, which treatment work best for people from different backgrounds, and by building a diverse database, which will play a crucial role in identifying further health condition and help as being the backbone for further research. My target here was to say if we need long read or not in this research. But first, to achieve the goal of All of Us, we need a really high number of samples, which actually they did. The project sequencing around 2 million samples using chip array. Subset of those was around 1 million was using short reads, and another subset for long read, but for research only. And the question I was asked here, do we really need long read? Aren't short and chip array is sufficient? So to answer this question, I took the genes that I mentioned earlier, the 386 that were identified as complex, and we tried to analyze them using short and long reads and see if we are able to solve them or not, meaning if long read is necessary to solve them. And these are the three questions that I asked. Are we able to identify SNVs, insertion and deletion, in repetitive region? Are we able to call a structural variant accurately? Are we able to detect epigenetics, which is a heritable phenotype that could cause diseases, but it does not involve any DNA sequence alteration? To summarize what we found, the conclusion of the analysis of short and long reads specifically on the medically relevant gene, we found the following. We could identify the SNVs and enders with high accuracy exceeding 99%. We identify structural variant with around 93% accuracy, which both are not achievable by short reads. We developed also methods and scripts to harmonize merge of different tools and algorithm. And we put all of this as a pipeline, cloud-based pipeline, to adapt our analysis for further research. And we put all of this in a paper called Utility of Long Read in all of us. So far, I believe I convinced you the importance of long read, and we managed to use them for rare diseases and also for medically relevant complex gene. So technically, is this the end now? Actually not. This has started another journey. While long read is crucial and important to solve both rare and complex diseases, and also to solve medically relevant complex gene, meaning now we are able to close the gap. However, it comes with a price. Not all laboratories have access to long read because of low budget or limitation. So my second quest here is to be able to make it affordable, meaning if I want really to pave the way for personalized medicine, I need to make this data or this technology available for small labs or labs with low budget. And this is what I have tried to do here, to extend what we learned from all of us by building a special kit that targets specifically challenging gene that needs long read to solve them. Not only that, I developed workflow and algorithm to actually reduce the burden or make it easy for laboratory with low or no expertise in bioinformatics to solve these cases. Now we are able to look to this gene. So if we have a researcher or a laboratory only focusing on this complex gene, we could help him in that. Alternatively, if we have a larger project that was based on short read and we need to complement them to solve what are not solved by short read, we can use also the kit I'm developing at the moment. To achieve that, I aligned with Twist and BagBio. Twist for capturing the region or the genes actually that are complex, and BagBio to support us with long, accurate reads so we are able to solve them. We included actually 389. I know that I have been repeating 386, but actually I added 3 notorious genes to our list to be able to solve them. We found that around 25% of the genes that we have at least one pathogenic variant from CLAN-VAR. We also expect this number should be higher, but because of their complexity, they have not been routinely analyzed. Thus, we have a low number. So if we are using long read further or this kit, we will be able to solve or to find more of these genes. As I have been talking too much now about their complexity, what did I do? So by using this kit, now we can multiplex or sequence 12 samples per one SMRT cell. Like normally we use at least 1 or 2 SMRT cells for whole genome sequencing. The kit is now able to do 12. But what about the coverage? Are we able to cover this complex medical genes? Actually, the answer is yes. Overall, we have an average coverage of 69x per gene. If we look to figure A, where we're here comparing the normalized coverage between the panel, where we are sequencing 12 samples, to whole genome sequencing from long read or from Illumina, where we are using one sample per smart cell, as you can see, we are surpassing their coverage, which is great, and we are reducing the cost 12 times. Additionally, the GC and EG content did not affect our average coverage. As we look into figure B here, we still have a high average coverage, even we have a sparse distribution of GC content. However, there are 4 genes that were not covered and actually used a different workflow to solve this gene. Beside coverage, I promise that I will be developing a computational workflow to make it easy for lab with no expertise or limited expertise in bioinformatics to solve this gene. In this pipeline, we use the modified reference, modified version of reference GHR38, in which they are trying to address the misrepresented region of the genome, including collapsed or duplicated region. Doing that will reduce the false positive rate of our result, and we are showing that later. Additionally, we used both mapping and assembly for variant calling to get the best of both worlds. So at the end, we have phased SNVs and SVs and indels. And for the genes that I mentioned that were not covered in the first slide, we used different tools to analyze them. And when we mentioned that they were not covered, we mean that their mappability was low because of difficulty in coverage. Not there is no read in there. So using this tool helped us to solve them. Now I'm showing you the workflows that we developed in a metrograph where we have different routes using mapping, aligning, and different methods to solve them. And there's a color for each band or each route. Starting by the green one, which represents mapping, where we have each step as an open circle. And then there are steps that we are branching to use also assembly in yellow beside mapping. Further downstream of this metro plan, we could see that we use the different tools like PharmCAD, Paraphrase, and Bango for solving the gene that we couldn't solve earlier with mapping and assembly, and annotate the data set. The result is converging at the end, as you can see, and filter them based on the allele frequency. And finally, you have phased SVs and SNVs in very coherent way and easy to run pipeline. That's all great. But what about the result? Are we sacrificing the result by multiplexing different samples? Actually not. Looking to the benchmarking of single nucleotide variant and indels, we could see that the panel achieved an accuracy of around 97, which is equivalent to what we could get from actually long-read whole genome sequencing, except with a fraction of the price. And we are surpassing what we could achieve by short-read Illumina. These values actually increase when we are looking only to exome. What about the structural variant? For the structural variant, as you can see here, we expected that Illumina in yellow will have a low performance because of the read length itself and hard to map, so there is no surprise here. However, if we look to the panel in blue and the whole genome sequencing long read in red, we could see that the result is similar, except that we are saving a ton of money, which means that we will be able to actually sequence more data. We wanted to prove that this was not by chance. So we replicated the analysis, meaning we did the same sequencing more and more. As if you look to the upper right figure, we have the panel 1, 2, 3, meaning that we have 3 replicates, and we compare them to benchmark data set, whole genome hi-fi, and whole genome Illumina. Looking to the blue bar, which is representing what could be the SNVs and indels that could be called by any method, short or long read, we find that around 78% could be called by all of them. However, looking to the yellow bar, which have around 3.2, this variant are only called using long read, either from whole genome sequencing or from the panel. Meaning that if we used short read, we are sacrificing around 3% of the data that are true variant, and we will not be able to call them. What is interesting and a really great thing, if we look to the green bar, which are around 0.7 or 1%. It's still around 400 variant, by the way. These variants are only called by the panel. That is because, if you remember, we used both mapping and assembly, so we managed to call variant even whole genome sequencing are not able to do that. Moving to structural variant, the lower graph on the right-hand side, the blue bar, which is around 17%, represents structural variants that we could call by long or short reads. The yellow one represents 37% or more, which is need long read to be called, either from whole genome sequencing or from the panel, which is now stressing and underscoring the importance of long read and how we could do that with the panel without sacrificing the price. I did not stop here, actually. I was a little bit greedy. So far I mentioned that we managed to sequence 12 samples per SMRT cell. The next question is, are we able even to sequence more than that? This is what I tried to answer here in this graph. If we look to graph 8, we did a down sampling. The blue one, which is 100%, meaning 12 samples. The orange one, it's 70%. Green one, 50%. Red one is 25%, and last one is 10%. Meaning we diluted the samples that we have to see how far we could go in sequencing sample per one SMRT cell without sacrificing the accuracy. If we started by SNVs here, you could see that we go to 25%, which represent 48 samples, without sacrificing SNVs. Same for SCVs. However, if we went to 10%, we see that there are a little bit drop in SNVs, so we say that we don't advise using more than 48 samples per SMRT cell. Same for SCVs, we still have the same performance. Even when we look at each gene separately in B graph, where we have box plot between 100 down samples, 70, 50, 25, and 10, we still see that the median in blue and mean in red is similar between different coverage. So from this slide, we could conclude that we could go down to 48 samples without sacrificing accuracy for calling SNVs and the unstructured variant, which is really great, and try here to make the kits that I'm suggesting as affordable as possible for different laboratory. By the success we achieved so far, we applied the kit that we have into two groups. The first one was Mendelian cases. If you remember, rare cases, 72% of it are Mendelian. So we was trying to see if we could use the kit to solve these cases and also with a good cost. We sequenced 3 unsolved Mendelian cases, like 9 samples. We identified the Mendelian concordancy between SNVs and SCVs. It was more than 96% and 94% respectively. We managed to identify de novo variant and autosomal recessive inheritance, as we are showing here in the IGV, where we have a variant exist in the mother and the father in heterozygous fashion. However, in the son, it presents in homozygous. So that could actually knock out the gene, which could explain this case. Nevertheless, the work here is still in progress, so this is inconclusive. Besides that, we also identified, applied the sample in cardiovascular cases, where we have 14 samples from different backgrounds, 12 European, and 2 samples from, 12 Hispanic and 2 European. Oh, sorry, my bad. I was right the first time. It's a 12 sample from European origin and 2 from Hispanic origin, my bad. And we was focusing on the LBA gene and the Kringle4 exon 2, which is responsible or related to the LBA concentration. As you can see here in LBA figure, we have the IGV. We managed to align and haplotype variant, as you can see in color brown and blue. We have 2 humps here, which represent the copy number variant in the Kringle4 exon 2 region, which we managed to identify. Additionally, we could see the correlation between the LBA coverage and the LBA concentration, which is reverse or negative coverage, negative relationship. As you increase the coverage, you have a higher LBA concentration. So we could here see that the kit is important to solve some cardiovascular cases. Besides that, we identified deletion of 82 base pair, which was systematic in chromosome 22 across all the sample. Even one of them are healthy, which explained that the lack of annotation and needing to focus more on structural variant calling using long read. Plus that there was samples that we identified a structural variant in JONX3 gene, which was recently related or linked to cardiovascular diseases. Finally, in this part, I would like to summarize what we managed to apply so far. We managed to accurately and cost-effectively capture the panel of the complex medically relevant gene, and we developed the workflow for it. We can now multiplex around 48 samples, reducing the cost needed to analyze genes, specifically with using long read, which make it now affordable for different laboratory. We did not sacrifice any of the accuracy of the SNVs and structural variant, where the result was equivalent to what we could achieve using whole genome sequencing from long read, around 97% for substitution and 94% for structural variant. We applied the panel in different scenarios, including Mendelian cases and cardiovascular cases. Work still in progress in this part. However, we have promising result. We put this work together and it's now accessible through medical archive. So I would encourage you, of course, to go through it and feel free to contact me if you need anything. Finally, I would like to thank my lab, specifically Fritz, and thank my colleagues and people that I have been working on. And thank you all for your patience and joining me in this talk. Feel free to reach me out through X or mail if you need any help or you are looking forward for cooperation. With that said, I will be more than happy to answer your question and inquiry. Thank you.
All right. Well, thank you so much, Medhat, for your informative presentation. We will now start the live Q&A portion of the webinar. So to our audience, if you have a question that you'd like to ask, please do so now. Just submit a question in the Ask a Question box located at the bottom of your screen. All right, so let's get started. The first question that I have for you asks, have you incorporated the CHM13 or T2T reference into your analysis?
Okay. T2T or CHM13 did a great work in solving some structural variant that we will not represent it in CHR38 before. However, it's still lacking the annotation. So while it's promising, but without an annotation applying it will be hard to resolve. So we could identify variant, but we will not be able to pinpoint what is the function of these variants.
Great, thank you. All right, next question here. What are the reasons behind not opting for long-read sequencing as the initial step in clinical whole genome sequencing?
Okay, that's a great question. So first of all, what comes to my mind is the cost. If you can solve it by a cheaper way, that would be first. So there are some things that we need to do first. First thing is start with, as you said, exome sequencing. If it's not solving it, yeah, we can go further. But if it did, then you save the money and still be able to do it. If this does not succeed, you go for whole genome sequencing. And in this case, long read will be the answer. However, again, I don't know if it's affordable for all labs or not to start with in the first place. Thank you.
Great, thank you. Now, you just mentioned cost, and this next question is asking what cost savings could be achieved by utilizing the panel approach compared to whole genome sequencing?
Okay, that's a nice one. So while the prices are changing, like now the price for both Oxford Nanopore and backbite is dropping, still, maybe high for some, but still dropping. Nevertheless, in this kit, as I mentioned, either you will reduce it by 12 folds if you just use 12 samples per smart cell, or you could go even to 48 fold reduction in cost if you use the 48 one. So in any ways, just use a proportion here. So I will not use the price because it's changeable, but you could reduce it from 12 to 48% lower than what you ever will do if you're sequencing whole genome sample in one smart cell.
Right, thank you. Well, it looks like that's all the time we have for today, so that will conclude our session. Medhat, do you have any final comments for our audience?
No, thank you, and feel free, if you're still in the in lab route here presentation, feel free to drop a question. If I'm not here, I will just write it on Twitter or whatever, and I will be answering you. I will be more than happy to answer all your questions now or later. I believe this This will be recorded, so even though if you saw it later, feel free just to reach out.
Yes, absolutely. Well, we want to thank you again, Medhat Mahmoud, for your time today and your important research. And before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 1 year until May 15th, 2025. LabRoots will alert you via email when it's available for replay, so we encourage you to share that email with your colleagues who may have missed today's live event. So until next time, take care, everyone. Goodbye.
Thank you.